Good day, everyone. I am Arithea Encarnacion, and I am one of the members of Group 4. And for today, we will discuss to you point and chromosomal mutations. Okay, so our agenda for today is listed here. First, we are going to discuss what is a mutation, the types of mutation. Next is what is point and chromosomal mutations, as well as its types. We also have examples of diseases caused by point and chromosomal mutations, and of course, we will discuss the similarities and differences of point and chromosomal mutations. Here we go. Ano nga ba ang mutation? So, mutation is a change in a sequence of bases in DNA or RNA. It is the change within a nucleic acid. So, mutations can result from DNA copying mistakes made during cell division, exposure to ionizing radiations, exposure to chemicals called mutagens, or infection by viruses. So, mutations are essential for evolution to occur. They are the ultimate source of all new genetic materials, new alleles, in a species. So, although most mutations have no effect on the organisms in which they occur, some mutations are beneficial. Even harmful mutations rarely cause drastic changes in an organism. Mutations can also be harmful or helpful. Beneficial mutations may become more common through natural selection, and harmful mutations may cause genetic disorders or cancer. Now let us talk about the types of mutation. The two main types that we are going to tackle is the point and chromosomal mutations. So, point mutation is a change in a single nucleotide in DNA. This type of mutation is usually less serious than a chromosomal alteration. So, an example of a point mutation is a mutation that changes the codon UUU to the codon UCU. Point mutations can be silent, missense, or nonsense mutations. Next is the chromosomal mutation. So, chromosomal mutation are mutations that change chromosome structures. So, they occur when a selection of a chromosome breaks off and rejoins incorrectly or does not rejoin at all. There are possible ways these mutations can occur. First is deletion, duplication, inversion, insertion, and translocation. So, chromosomal alterations are major changes in the genetic material and very serious. They often result in the death of an organism in which they occur. So, if the organism survives, it may be affected in multiple ways. An example of a human chromosomal alteration is a mutation that causes Down syndrome. It is a duplication mutation that leads to developmental delays or other abnormalities. These are the other three types of mutation: the germline mutation, frameshift mutation, and somatic mutation. Germline mutation occurs in the gametes. These mutations are especially significant because they can be transmitted to the offspring, and every cell in the offspring will have the mutation. Somatic mutation occurs in other cells of the body. These mutations may have little effect on the organisms because they are confined to just one cell and its daughter cells. Somatic mutations cannot be passed on to the offspring. And the frameshift mutation is a deletion or insertion of one or more nucleotides that changes the reading frame of the base sequence. Deletions remove nucleotides and insertions add nucleotides.
So my name is Roger Stewart Dahl, and I will be discussing about the point mutation. So point mutation is an alteration in a single nucleotide in a DNA or RNA sequence. So it can happen due to changing, inserting, or deleting a single base pair in the nucleic acid. So most point mutations can occur due to the errors in the DNA replication process. So furthermore, point mutations can also take place due to some other reasons such as exposure to UV or X-rays and carcinogenic chemicals. So, um, based on the effect of the final product, point mutations can be further grouped as silent mutations and missense mutations and also nonsense mutations. So, silent mutations result in functionally same amino acids at the end, while missense mutations result in functionally different amino acids. So the resultant protein in missense mutation may show alteration or loss of function accordingly. So nonsense mutations cause premature protein synthesis, which leads to the complete loss of protein function. And lastly, point mutations can cause several diseases, including cystic fibrosis and sickle cell anemia, and also Tay-Sachs disease. So the next topic is types of point mutation, and which will be discussed by my group.
Good day, I am Mary Joy Delgado from class MLS2C, and I am gonna discuss the type of mutation, point mutation. Types of point mutation. There are two types of point mutation. First is the substitution, and second is the insertion and deletion. So a substitution mutation occurs when one base pair is substituted for another. So for example, this would occur when one nucleotide containing cytosine is accidentally substituted for one containing guanine. And there are three types of substitution mutations. So first is the nonsense. It occurs when one nucleotide is substituted, and this leads to the formation of a stop codon instead of a codon that codes for amino acid. So second is the missense. So it occurs when one nucleotide is substituted, but in a different codon is formed. So this time the codon that formed is not a stop codon. And the last one is the silent. So a nucleotide is also substituted, but the same amino acid is produced anyway. And this can occur because multiple codons can code for a same amino acid. The second type of point mutation is the insertion and deletion. So an insertion mutation occurs when an extra base pair is added to the sequence of bases. So the insertion we can see at my side. So that is in the center, that is the insertion.
So good day everyone, I am Eileen Tablanda and I'll be reporting the examples of disease caused by point mutation. So in point mutation we have three examples. First is the sickle cell anemia, second is one form of beta thalassemia, and third is cystic fibrosis. So first, we have the sickle cell anemia. So sickle cell anemia is one of a group of disorder known as the sickle cell disease. Sickle cell anemia is inherited red blood cell disorder in which there aren't enough healthy red blood cells to carry oxygen throughout your body. So the type of mutation is substitution, and the description is one base incorrectly added during the replication and replace the pair in the corresponding position of the complementary stand. So second is the one form of beta thalassemia, which is classified into two types of depending on the severity of symptoms: thalassemia major, also known as Cooley's anemia, and thalassemia intermedia. So the type of mutation is insertion, which is one or more extra nucleotides are inserted into replicating DNA, often resulting in a frame shift. So we are here in the last example of disease caused by point mutation, which is the cystic fibrosis. It is an inherited disorder that causes severe damage to the lungs, digestive system, and the other organs in the body. And the type of mutation is deletion, and it means one or more nucleotides is skipped during the replication or otherwise exists, often resulting in a frame shift. So that's all for the example of disease caused by point mutation.
So, good day everyone. I am Kiara Eliza O. De La Cruz, and today I will be the one who's going to discuss about the chromosomal mutation. So, firstly, do you have any idea about this topic from our previous reporters? I hope you do, but I guess you also have. And I know that you are listening to them as well. So I hope you're going to listen to me. So, firstly, what is chromosomal mutation? So, this is a mutation involving a long segment of DNA. So, these sections of the DNA can be deleted, inserted, or it can also be inverted in these alterations. So, also in some situations, the deletions of chromosomes can attach to other chromosomes. So, causing this would trouble both the chromosomes that lose and gain a DNA. So, a chromosomal arrangement is another name for this kind of mutation. This mutation as well will affect the entire chromosomes and the genes, not just the individual nucleotides. So this mutation will occur when a part of the chromosome will break off. So it gets duplicated or it moves to another chromosome due to the faults in cell division. This will change the structure or the number of the chromosomes. So if you want to know more about this topic, which is the chromosomal mutation, please do listen to our next reporter, because the next reporter will discuss about the types of this kind of mutation. So thank you and God bless.
Hello, I am Ganat Sarpamanas. Now that we've discussed about the definition of chromosomal mutation, let's proceed with the types of chromosomal mutation. The types of chromosomal mutation can be broadly categorized into two groups, namely chromosomal mutations 1 and chromosomal mutations 2. Chromosomal mutations 1, or structural chromosomal instability, has four types, namely inversion, deletion, duplication or amplifications, and translocation. Chromosomal mutations 2, or numerical chromosomal instability, has two types, namely aneuploidy and polyploidy. For chromosomal mutations 1, or structural chromosomal instability, let's talk about inversion first. Inversion is a type of structural mutation where a part of chromosomes or a set of genes rotates by 180 degrees on its own axis. There is no net loss or gain of genes, but simply a rearrangement of the sequence. A part of the chromosome is broken and then rejoins in a different direction. Inversion mutation can be detected cytologically in the meiotic nuclei by the detection of an inversion loop in the paired homologs. If identical inversion occurs in each member of the homologous chromosome, normal distribution of the chromosome occurs. However, inversion heterozygotes might produce deletions and duplication during crossing over. For the types of inversion, we have paracentric inversion and pericentric inversion. If the inversion doesn't include the centromere, it is called paracentric inversion, while if the centromere is included, it is called pericentric inversion. For the second type of chromosomal mutations 1, we have deletion. Deletion is a type of structural mutation that occurs due to the loss of a part of a chromosome as a result of the breakage of the chromosome. usually due to lag during anaphase and digestion by nucleases. Chromosomes that have undergone deletion cannot revert back to normal, and if transmitted to the next generation, can be hereditary. Deletion is lethal in the case of homozygous chromosomes, and even in heterozygous chromosomes, there are slim chances of survival. Heterozygous chromosomes can, however, survive smaller deletion. Now for the types of deletion. Deletion can either be terminal or intercalary. The terminal deletion occurs due to the loss of the terminal section of a chromosome. This involves a single break in the chromosome. The intercalary deletion occurs due to the loss of an intermediate section of the chromosome. This involves two breaks on either end of the deleted section. The third type is duplication or amplifications. Duplication is a type of structural mutation where a part of a chromosome is present in excess of the normal composition. The genes present in a cell might exist in more than 2 doses as a result of duplication. The fourth one is translocation. Translocation is a type of structural mutation resulting from the shift or transfer of a part of a chromosome or a set of genes to a non-homologous chromosome. There is no net gain or loss of chromosomes or genes during translocation, but a rearrangement. Now, there are 3 different types of translocation depending on the pattern of rearrangement: simple translocation, shift translocation, and reciprocal translocation. Simple translocation involves a single break in the chromosome where the broken piece then attaches to one of the ends of a non-homologous chromosome. Shift translocation involves the insertion of a broken segment of a chromosome interstitially in a non-homologous chromosome. Reciprocal translocation is the exchange of a translocated segment with another in order to create 2 translocated chromosomes. Now for chromosomal mutations 2 or numerical chromosomal instability. Chromosomal mutations 2 include mutations that are caused by the alterations in the number of chromosomes in a cell. The change in the number of whole chromosomes is called heteroploidy. It produces phenotypic changes, modifications of phenotypic ratios, and alteration of linkage groups. Heteroploidy can be further divided into 2 different categories depending on the changes in the entire set of chromosomes or in the single whole chromosome. The first category is aneuploidy. Aneuploidy is a type of mutation that changes parts of a chromosome set, resulting in either the loss of one or more chromosomes or the addition of chromosomes. Aneuploidy resulting from the loss of chromosomes is called hypoploidy, whereas that due to the addition of chromosomes is called hyperploidy. Hypoploidy usually occurs due to the loss of a single chromosome or monosomy, or due to the loss of a pair of chromosomes or nullisomy. Hyperploidy in turn might involve the addition of a single chromosome, now then called trisomy, or the addition of a pair of chromosomes, tetrasomy. Aneuploids are caused as a result of non-disjunction or the failure of the separation during mitosis or meiosis. The second category is polyploidy. Polyploidy is a type of euploidy where an organism has more than 2 sets of genomes. Polyploidy includes different combinations like triploid, tetraploid, pentaploid, hexaploid, and octoploid. Polyploidy can be further divided into 3 groups, namely autopolyploids, allopolyploids, and autoallopolyploids. Autopolyploids are polyploids that consist of the same basic set of chromosomes but multiplied to form multiple sets. Allopolyploids are the polyploids that result from the doubling of chromosome number in a hybrid from 2 different species. The next topic will be the examples of chromosomal disorders to be discussed by Ms. Jupeka Zapanta.
Hi, I am Jabeka Zapanta and I will be presenting the examples of disorders caused by chromosomal mutations. We have deletion, duplication, inversion, translocation, aneuploidy, and polyploidy. For deletion, we have cretochat. It is a chromosomal condition that results when a piece of chromosome 5 is missing. Often have a high-pitched cry that sounds like that of a cat during infancy. Second is we have the George syndrome. It is a disorder caused when a small part of chromosome 22 is missing. This deletion results in the poor development of several body systems, including heart defects, poor immune system function, a cleft palate, complications related to low levels of calcium in the blood, and delayed development with behavioral and emotional problems. For duplication, we have first is the Charcot-Marie Tooth Disease Type 1A or CMT1A. It is a duplication of chromosome 17 and it is a type of peripheral neuropathy, a condition affecting the transmission of information between the central nervous system, such as the brain and the spinal cord, and the rest of the body. Next is Pallister-Killian. It is a result of extra chromosome 12 material. Babies with this syndrome may have many problems. These include intellectual disability, poor muscle tone, coarse facial features, and a prominent forehead. For inversion, we have Ambras syndrome. It is the inversion of chromosome 8 and a very rare type of hypertrichosis lanuginosa congenita, a congenital skin disease characterized by excessive hair growth on the entire body, with the exemption of the palms, soles, and mucous membranes. Next is we have Walker-Warburg syndrome. It is the pericentric inversion of chromosome 9. It is an inherited disorder that affects the development of the muscles, brain, and eyes. Affected babies have weak muscle tone or hypotonia, typically have a brain abnormality called cobblestone lissencephaly, in which the surface of the brain lacks the normal folds and grooves and instead develops a bumpy, irregular appearance. Eye abnormalities are also a characteristic of Walker-Warburg syndrome. For translocation, we have de la Chapelle syndrome or XX male syndrome. It is a condition in which individuals with two X chromosomes in each cell, a pattern normally found in females, have a male appearance. They generally have a small testes and may also have abnormalities such as undescended testes or cryptorchidism or the urethra opening on the underside of the penis or hypospadias. Next is we have translocation Down syndrome. Translocation Down syndrome is a type of Down syndrome that is caused when one chromosome breaks off and attaches to another chromosome. In this case, there are 21 chromosomes, but one of the 21 chromosomes is attached to another chromosome. The genetic material from the extra 21 chromosomes is what causes the health problems that are caused by Down syndrome. In the translocation Down syndrome, the extra 21 chromosome may be attached to the 14 chromosome or to other chromosome numbers like 13, 15, or 22. In some cases, there are 21 chromosomes that can be attached to each other. There are no big differences between the patients who have translocation Down syndrome compared to trisomy 21. For aneuploidy, we have Edwards syndrome. There is an extra copy of chromosome 18. Trisomy 18, also called Edwards syndrome, is a chromosomal condition associated with abnormalities in many parts of the body. Individuals with trisomy 18 often have slow growth before birth or intrauterine growth retardation and a low birth weight. Affected individuals may have heart defects and abnormalities of other organs that develop before birth. For polyploidy, we have partial hydatidiform mole. A partial hydatidiform mole is a triploid pregnancy with 69 instead of 46 chromosomes and another strange complication of the reproductive process. Partial hydatidiform mole appears to be the result of what happens when this cortical granule process fails and an egg can be penetrated by multiple sperms. It can be indicated by ultrasound showing placenta tissue, some hygromatocysts, and some fetal oddments, but no clear fetal structure.
Hi everyone, I'm Crystal G. Mahinay and I will be discussing to you the difference between point mutation and chromosomal mutation. But first, let us define what is point mutation and chromosomal mutation. So point mutation is a single nucleotide change that occurs in a DNA or RNA sequence, while chromosomal mutation is a structural or numerical change in chromosomes. So, thus, this is the key difference between point mutation and chromosomal mutation. So, in point mutation, changes occur in a single base pair, but in chromosomal mutation, changes occur in a part of a chromosome or the total number of chromosomes in a chromosome set. So, therefore, there is also a significant difference between point mutation and chromosomal mutation. Moreover, a further difference between point mutation and chromosomal mutation is the impact of the mutations. The impact of a point mutation is comparatively low while it is high in chromosomal mutation. Also, the point mutations are caused due to insertions, deletions, changes, and etc. So while chromosomal mutations are caused due to duplication, translocation, inversions, deletions, non-disjunctions of chromosomes, crossing over, and etc. Therefore, this is the major difference between point mutation and chromosomal mutation. So, sickle cell anemia, hemophilia, cystic fibrosis, Huntington syndrome, Tay Sachs disease, and cancers are some of the diseases that occur due to point mutation. So, on the other hand, Klinefelter syndrome, Turner syndrome, and Down syndrome are some syndromes that occur due to chromosomal mutations. In summary, point mutation refers to a single base pair alteration in a DNA or RNA sequence. So it is a small scale mutation, whereas chromosomal mutation refers to a structural or numerical change in chromosomes of an organism. So it is a large scale mutation. So the impact of chromosomal mutation is high since the altered segment of a chromosome may include several of many genes. So point mutation occurs due to change, insertion, or deletion. On the other hand, chromosomal mutation may occur due to duplication, translocation, inversion, deletion, non-disjunction of chromosomes, crossing over, and etc. So thus, this is the summary of the difference between point mutation and chromosomal mutation.
Hi everyone, my name is Haja Asajani and I'm going to discuss the last topic for this video, which is the similarities between point mutation and chromosomal mutation. So as you guys can see, there are only three similarities. So first, point mutation and chromosomal mutations are two types of genetic mutations occurring in a living organism. So when we say genetic mutation, it is a permanent alteration in the nucleotide sequence of one or more genes or in the number or structure of one or more chromosomes. Second, in both cases, the nucleotide sequence are altered. So what happens if your DNA is altered? When a gene mutation occurs, the nucleotides are in the wrong order, which means the coded instructions are wrong and faulty proteins are made or control switches are changes. The body can't function as it should. Mutations can be inherited from one or both parents. Lastly, both types of mutations can cause lethal disease. So lethal disease is a deadly disease. Mortal implies that death has occurred or is inevitable. So that is all, and I hope you learned something from our video. Thank you. Bye.